Dear students, welcome to Tungala online classes. Today I am going to start with a new chapter, that is cell cycle and cell division. Right? It's very important chapter for you people, and in NEET examination definitely one or two questions so are usually asked on this chapter, and a very important chapter it is. So majority of the questions are very tricky in this case, and you have to solve them with using some of the tricks and some of the numerical problems also you will come across on this chapter itself, right? So let us start with the cell cycle and cell division now, right? So what is cell division? The question arises. So when the cell, so that is going to form its daughter cells from the pre-existing ones, right? So the process of formation of daughter cells from the pre-existing ones is called as cell division, or it is also called as cell reproduction, right? Or cell multiplication it is also called as, right? So let us write it now. Cell division. It is nothing but the process of formation of daughter cells from the parent or pre-existing ones. Parent cell is called cell division, or it is also called as cell multiplication, or it is also called as cell reproduction. Cell reproduction, right? So this is more important aspect. So we came to know what is a cell division. Means it is very simple aspect. So here a single cell is there, and this cell has 2n number of chromosomes, that is diploid number of chromosome. It divides into two smaller sized cells, so they have 2n and 2n, equal amount of genetical material they contain, that is 2n number of chromosomes it also contains. But these two cells are said to be as daughter cells. These are called as, and this is called as parent cell. So the process of formation of daughter cells, here the process of formation of daughter cells from the parent cell or pre-existing cells. This is the parent cell or the pre-existing cell. So it is called as cell division or cell multiplication or cell reproduction it is called as, right? So what is the use of this cell division? It is one of the fascinating characteristic feature of the cell itself, right? It is one of the fascinating character of the cell. This cell division leads to growth of an organism. Means as a result of cell division, so the organism that grows, right? So this is the fundamental factor that you must remember over here. And so this cell division occurs in all the organisms and responsible for the growth of the organism, I said, right? So but one thing we must remember, that we are all formed from the single diploid cell itself. The life of each organism that starts from single cell, right? If a single cell is there, we might be the single celled organisms, but we are multicellular organisms today. So it is because of a single cell which can divide itself. So when a single cell cell start to divide itself, it becomes a numerous number of cells, and innumerable number of cells start to do certain functions. So within the body, being within the body, right? So that forms the entire organism itself, right? See here, the life of all organisms starts from a single cell. That single cell itself, consider it is zygote. That is nothing but zygote. Zygote divides and redivides us to form what? Embryo. Embryo that grows into what? Fetus. Fetus grows into what? A new individual. Right? And this new individual is multicellular in nature, and this is single cell. From the single cell, multicellular organisms have been made over here. This is the fact what you must be aware of that one. That's why cell division is must. It must take place in the organisms that leads to the growth of the organism. Growth and reproduction are the important features of the cell. Remember, growth and reproduction are important features of living organisms, and even the cell also. It is the characteristic feature of a cell, and simultaneously it is the characteristic feature of all the living organisms, right? So this is very important aspect to be remembered. And now we shall discuss what are the types of cell division. There are different types of cell division that we come across. What are those types of cell divisions? So let us go through. Types of cell division. So in how many ways the cell divides? So how many cells it is going to form as a result of a division of a single cell? See here, this is one cell which has a diploid number of chromosomes, that is 2n number of chromosomes. It divides into two equal sized daughter cells with the same genetical material or same number of chromosomes within themselves. So this is one type of cell division. And here you can observe equal number of chromosomes are shared between two daughter cells, and both the daughter cells are smaller enough when compared to the parent cell, right? So when compared to the parent cell, they are smaller enough, but still these cells have the same ability, the same potentiality what the parent cell has. These cells are also growing themselves. They grow and once again they can multiply themselves. So this is the characteristic feature of parent cell. The same characters are there with these two daughter cells also. Is it clear? Now it is very clear to you all, right? So this is one type of cell division. This is called as mitosis. This is called as mitosis or mitotic cell division. And another type of cell division is there. 1 or 2n number of chromosome having one cell is there, diploid cell is there. It divides into two equal sized daughter cells, but only the difference is half the number of chromosomes are there in the first division. See here, the same cell undergo the division twice. Means it undergoes division twice. Twice in the sense of 2 times. Means this cell divides into 2 equal sized cells, and again these cells divide into 2 equal sized cells. And all the cells have half the number of chromosomes of its parent itself. So how many number of chromosomes are there in this one? 2n. How many number of chromosomes are there here? It is N. N in the sense that haploid number of chromosome. Only one set of chromosome. Here two set of chromosomes are there. Two set of chromosomes when you consider human being, it is 46 and it is 23 in case of this one. Right? And this type of cell division is called as meiosis. What it is called as? Meiosis. How to define mitosis and how to define meiosis is the thing. What is mitosis? It is a type of a cell division in which a diploid cell divides into two equal sized or identical daughter cells with the same number of chromosomes or the same amount of genetical material what is there in the parent cell is called as mitotic cell division. And meiosis means it is a type of a cell division in which a diploid cell divides into, a parent cell divides into four equal sized cells having half the number of chromosomes or half the amount of genetical material within them respectively is called as meiosis. Shall we write the definitions? It is the kind of cell division in which it is a kind of a cell division in which a parent cell divides into two identical cells with same number of chromosomes or equal amount of genetical material. Equal amount of genetical material is called as what? Mitosis. What do you mean by meiosis? It is also a kind of cell division in which a parent cell divides into four equal sized cells with half the number of chromosomes or half amount of genetical material. Half the amount of genetical material within themselves. Such kind of cell division is called as meiosis, and this is called as mitosis. Mitosis is a type of cell division which take place always in what? Somatic cells, right? So this mitosis is always taking place in somatic cells. Somatic cells in the sense that they are body cells. Right? So cell division occurs in body cells, and this is meiosis. Meiosis always take place in so the cells which form the tissues. You write here tissues which form gametes. Right? Means during gamete formation meiosis occurs, during the body cell division mitosis occurs. So this is the fact what you must be aware of that. Okay. So remember here in case of mitosis, so equal sized two identical cells with same number of chromosomes are shared between two daughter cells, but here half the amount of genetical material or the half the number of chromosomes are shared among the four daughter cells. In case of meiosis, this occurs during gamete formation. Meiosis occurs during gamete formation. Mitosis occurs in somatic cells. Mitosis is occurring in lower organisms, that is unicellular organisms, so as to produce more number of cells from single parent cell. Right? It is also possible there. It is also possible there. Multiple fission is one kind of the mitotic cell division that occurs so as to form many number of daughter cells from a single parent cell by the division of the parent cell itself. Right? So that is the fact what you should be aware of this one. Right? Types of cell division these are. Okay. Next concept is, that is cell cycle. What is cell cycle? Let us see now. See here, one cell that duplicates itself, right? So during its duplication, it come across on orderly events. Orderly, some of the events are taking place continuously during the cell division itself, and that orderly sequence of events is called as what? Cell cycle. It is called as cell cycle. A cell perform an orderly sequence of events during its division is nothing but cell cycle itself. During cell cycle, what happens here is, here the duplication of the genetical information or the genetical material, that is DNA or the number of chromosomes that take place over here. So what initially take place? Duplication of genetical material and duplication of other cell contents also take place. That is cytoplasmic material accumulated itself, and even the genetical material that duplicates over here. So all together perform what? That is cell cycle itself. Right? So then how to define this cell cycle is the question arises here. Cell cycle otherwise said to be as, see here, one cell, that is a diploid cell, divides into two equal sized cells. This is the fact you know. Cell cycle always starts from here, from a single cell. It is a grown cell and it divides into two daughter cells, and respectively these two daughter cells grow themselves as if it's parent cell. Right? So from here to here, what is the sequence of events that occur over here? All these sequence of events, that is nothing but cell growth and division, and again growth of daughter cells. So this is totally called as what? Cell cycle it is. Totally it is said to be as cell cycle. Right? So now we came to know what is cell cycle. Whenever a cell grows and divides into two daughter cells, and respectively those two daughter cells, when they start to grow and mature themselves, so and ready to divide itself up to that level, so the cell cycle occurs over here. So this is the fact what you should know. Then how to define the cell cycle? Cell cycle. It is a sequence of events takes place during which the genetical material or chromosomes duplicate and other cell contents also to form two daughter cells. See here, what happens here? So it is the sequence of events that take place during the cell division. So where the multiplication of the genes or the genetical material that take place along with that one. So once again the cellular contents also duplicate themselves, and that leads to formation of two equal sized daughter cells. It's called as what? Cell cycle itself. It is called as cell cycle. So the time period for this cell cycle itself is different in different organisms, right? The time period of cell cycle is different in different organisms. Say for example, the time period that differs. In bacterial cell, in bacteria, it takes place 30 minutes to complete the cell cycle, right? So this is one fact. In case of human being, it is about 24 hours. It is about 24 hours. In case of yeast, it is 90 minutes. In case of yeast, it is 90 minutes, right? And 8 to 10 hours in case of mucosa cell of intestine is about 8 to 10 hours. It differs from one organism to another organism, right? And we come across even the generation time, right? What is generation time? It is the time period from the beginning of cell growth up to the cell division and once again the growth. So that period itself is said to be as generation time. What is generation time? It is the beginning of one cell division up to the next cell is called as generation time. This is more important aspect to be remembered. So generation time is very important here, right? So and this cell cycle has two main phases: cell cycle has two main phases. One is called as interphase, second one is called as M phase or mitotic phase. M phase or mitotic phase. So these two phases that we come across in the cell cycle. So this interphase is said to be as preparatory phase. It is said to be as a preparatory phase, and this one is called as a division phase. Right? So there are two phases that we come across in the cell cycle. So what they are, how they are, everything that we study now. Now the phases of cell cycle. What are the two major phases? One is called as interphase and second one is called as M phase. Are the two phases that we come across over here? One is called as interphase. What is this interphase? How does it take place? Let us see. So it is said to be as a preparatory stage. It is a preparatory stage. And it occurs in between two successive mitotic cell divisions. It occurs in between two successive mitotic cell divisions. Right? It occurs over here. And this interphase usually, see here, two phases usually in case of human being. In human being, interphase occurs about 23 hours. Already I told you, one cell cycle of the human cell that take place 24 hours. In that 24 hours interphase take 23 hours, and M phase, that is mitotic phase, take place only 1 hour. See here, 95% of time has been taken by this interphase, only 1 hour is taken by this M phase. Means this plays a very important role over here. In this case, in interphase, so all the active growth is taking place, or the duplication of the materials is taking place during this period. So during this period, active growth and duplication of materials is taking place during interphase. So highly intense active cell growth as well as duplication of the materials is taking place in this interphase. But in case of M phase, it simply divides, the cell divides. So let us see diagrammatically how this cell cycle is. Right? So this is how we are going to show the cell cycle over here. This interphase is divided into three phases. One is gap 1 phase, G1 phase, S phase, and G2 phase. So let us see them here also. So this is G1 phase, okay, and this is the central point from which you can write here G2 phase, G1, sorry, it is S phase, and this one is G2 phase, and this is M phase. After M phase, there we come across Cytokinesis. Cytokinesis, cell division occurs over here. Here you come across some M phase, that is mitotic phase. See here 4 stages that you come across: 1, 2, 3 and 4. So this is the phase called as, prophase. This is metaphase, this is anaphase, this is telophase. Okay, there are 4 phases that we come across over here, right? After that one, it is followed by cytokinesis. And one more thing you must remember over here: after cytokinesis, there you come across a small period, that is G0 phase. G0 phase is the resting period of the cell. This is how the cell cycle that take place over here. Alright? See here, from this one up to this one, from here to here it is interphase. Here it is interphase. From here to here it is M phase, and here to here it is cytokinesis, and here to here it is G0 phase. So this is how the cell cycle that occurs over here. So first of all, we'll go through the interphase part, and in interphase, once again we divide the period into 3 periods or 3 phases itself, and those phases are gap 1 phase, S phase as well as G2 phase. What happens actually during these 3 phases? So let us study first, and afterwards we'll go through what is M phase, how does it occur, and cytokinesis as well as G0 phase. All these are the short explanations we are going to study over here. And so later, during the study of mitotic cell division as well as meiotic cell division, we study in detailed manner the other things over here, right? In interphase, the first phase is G1 phase. Let us start with the G1 phase first. In interphase, there are 3 phases that we come across here. One is G1, and second one is S, and the third one is G2 phases. So let us start with the first one, that is G1 phase, or this is called as gap 1 phase, or this is called as post mitotic phase. It is called as post mitotic phase, or it is also called as pre-synthetic phase. Pre-synthetic phase, first growth phase. These are all the names for the G1 phase. These are all different names for G1 phase itself. So pre-synthetic phase, why it is called as, is because the synthesis of the genetical material that take place in S phase usually. That's why before the S phase, this phase is there, hence it is called as pre-synthetic phase itself, right? So let us see what happens over here. Here in this case, the cell will always be metabolically so active, and cell growth is continuously taking place over here. So the cell will be metabolically very active, and cell growth is taking place over here. So during this stage, what happens over here is, so here the intensive growth of cellular components is taking place. Intensive growth of cellular components, many cell components except DNA is taking place over here in this phase also. So cellular components except DNA or the genetical material, remember, right? Here then what happens here, the duplication of or the production of cell organelles take place. So organelles like what? Mitochondria is there, chloroplast is there, lysosomes are there, right? So there the ribosomes, and apart from this one, mRNA, tRNA, rRNA, all these are also taking place simultaneously. So all these RNAs are produced over here, and along with this one, some of the structural and functional proteins are produced over here. Here some of the structural and functional proteins are produced over here. So, and the next one is very unique one. See here, some of the substances are produced here in this phase so as to continue in the next phase, right? So that stimulate or that may inhibit the continuation or the inhibition of the next phase itself, right? So there are some substances produced during G1 phase so that regulate the proceeding or inhibition of next phase, and such kind of regulatory proteins are there, and those regulatory proteins are produced whether, so these proteins that stimulate the next phase, so going to the next phase, or inhibit the next phase, so that is done by some of the regulatory protein called as protein kinases. And these protein kinases are stimulated by cyclins. What are those one? So they are cyclins, right? So protein kinases are of different types. So cyclins are there, and hence these are called as cyclin dependent protein kinases. They are simply called as CDK. Yes, CDKs they are called as cyclin dependent protein kinases. These are called as CDKs. These CDKs are specific for a specific phase, right? So this is the fact what you must remember. See here what happens here. Specific means example G1 phase is there. For this phase G1 specific cyclins are produced. G1 specific cyclins are produced here, and these cyclins, so they stimulate these CDKs and they regulate G1 phase. In the same way, S phase is there, and S specific cyclins are produced and they stimulate the S phase itself. Like this. So these are specific proteins that stimulate the process in respective phases itself, right? So these are the things what you must remember over here, right? Next one is very simple one. What is that? Is, see here, G1 specific cyclins are there. Here protein kinases are produced over here, and these protein kinases, so they are depending upon what? So these are depending upon, or for their function they depend upon these cyclins. Hence these are called as cyclin dependent protein kinases, and these, so they help in going to the next phase. So they stimulate each phases there from G1 to S phase. If they want to go, then they depend on CDKs itself. This is the fact what you must remember in this case. And one more thing, so in this case is, these specific cyclins are destroyed over here after one particular phase, right? Say for example, if G1 phase is completed, then G1 specific cyclins or CDKs of G1, they have been destroyed, and the next one starts the S phase. So the S specific cyclins are produced, they continue the S phase, and so they also destroy after S phase, that go to the G2 specific cyclins are produced, and ultimately after the G2 phase, so they also destroyed over here. This is the fact you must be aware of this one, right? And after this one, let us go to the next phase, that is S phase itself. Let us start with S phase. Second phase is S phase. It is called as synthetic phase. In this phase, what happens here is, here the replication of the major function is replication of DNA that take place, or the synthesis of new DNA that takes place over here in this period, and the proteins associated with the DNA are also synthesized in this phase. So those proteins are called as histone proteins. These histone proteins are also produced there during this phase, and DNA is also synthesized in this phase, and the amount of DNA that produced in each cell itself, that doubles. Say for example, DNA of a diploid cell, diploid cells that duplicates from 2C to 4C, that increases 2C to 4C, and the DNA of a haploid cell is about 1C that duplicates to 2C. Double the amount of the genetical material that take place over here, and the number of chromosomes also duplicate longitudinally. So say for example, see here, number of chromosomes does not increase here as a result of the cell, as a result of duplication of this DNA material. See here, if this is one chromosome, this chromosome so divides into two equal halves so as to form this kind of chromosome. Now the genetical material is increased from 1C to 2C in case of haploid cell, 2C to 4C in case of diploid cell, of course. So it is duplicated. It is duplicated. But the number of chromosome is same. Here also only one number and here also only one number. And this type of chromosome itself is called as dyad. It is dyad means the chromosome with two chromatids. Two chromatids. The chromosome with two chromatids. Here the chromosome with only one chromatid that divides into two halves so as to form dyad condition. Right? So as to form dyad condition. But the number of chromosomes does not increase over here. That is why during the mitotic cell division what happens over here is a number of chromosomes that are equally shared in both the daughter cells. Both the daughter cells. What are the number of chromosomes that are here? So when the DNA that duplicates, so that is 2n, 2C it is, and this 2C increases to 4C, and once again 4C is divided into 2C itself, right? So once again in daughter cells the same number of chromosomes or the same amount of genetical material that remains there itself. So this is about the synthetic phase, right? One more thing we must remember regarding this S phase, what is that is the spindle fibers. The spindle fibers what are formed from the centrioles. These centrioles they duplicate themselves. They duplicate themselves during this phase. Centrioles duplicate themselves during this phase. Alright? So that would help in the synthesis of these spindle fibers, of course. Alright? So here chromosomal number is not at all duplicated. Right? Not at all increases to double the number of chromosomes. The number of chromosomes will be same, right? But the centrioles that duplicate here in S phase, right? So this is lasting for about 8 to 9 hours, and G1 phase is lasting for about 10 hours to 12 hours sometimes. Means maximum time has been taken by the G1 phase you are seeing over here. So maximum 10 to 12 hours, it is 8 to 9 hours, is 2 to 5 hours G2 phase that take place. So like this, so this is only 1 hour that take place over here. So this is 23 hours, this is only 1 hour that take place over here, right? So these are the facts what you should remember regarding the G1 phase as well as S phase. And the next phase itself is G2 phase. What happens in case of G2 phase is, so there also the cellular components once again duplicate themselves. Say for example, the cell components or the cell organelles like mitochondria as well as chloroplasts, all of themselves they start to duplicate and produce more and more in number itself, right? Along with this one, some of the proteins are also synthesized in this stage itself in the G2 phase. It is also called as Cap2 phase. In this phase, what happens over here is, here the duplication of or production of once again cellular components that takes place over here, especially in mitochondria as well as chloroplasts are duplicated by binary fission. They produced their result. Along with this one, some other components like tubulin protein is also produced over here, and as a result of this tubulin protein production, there the microtubules are produced over here, and these microtubules are going to form spindle fibers here. Spindle fibers here. These spindle fibers are very important, right? So they are made up of microtubules. These microtubules are made up of the protein called as tubulin protein. There the tubulin protein is produced much there itself, right? In G2 phase. Cellular components, many number of proteins are also produced over here. Protein synthesis also take place over here, so as to produce many structural proteins here in G2 phase. So this is all about G1 phase as well as G2 phase as well as S phase. These three phases are long lasting one, and so in that G1 phase is taking maximum time period over here, right? And after that one, genetical material is duplicated here. Even centrioles are duplicated over here. Here once again mitochondria, chloroplast are produced over here. Once again protein synthesis is taking place here, and even the spindle fibers produced over here. All these are the activities that are taking place in each phases of the interphase itself. Come to the next phase, that is M phase. M phase. M phase is mitotic phase. So this lasts during in case of human being, this lasts for about 1 hour, right? This is M phase. So during this phase, what happens? This phase is the important phase during which the division of the nucleus occurs first, and later division of cytoplasmic contents that take place over here. The division of the nucleus occurs first, and the division of nucleus is called as karyokinesis, and this is called as cytokinesis. Cytokinesis. Kinesis means breakdown. Breakdown of what take place? Karyon. Karyon means what? Nucleus. Nucleus breakdown is nothing but nuclear division, and the division of the cell it leads to, right? So this M phase is divided into division of the nucleus and the division of cytoplasmic content, that is karyokinesis is the first step and cytokinesis is the second step. So during karyokinesis, so there we come across 4 phases. There we come across 4 phases. Those are prophase, metaphase, anaphase, as well as telophase are the 4 phases that we come across in this stage. So prophase, metaphase, anaphase as well as telophase are the 4 phases that we come across in karyokinesis. After this one, after telophase, it is followed by cytokinesis. Cytokinesis over here, right? So that is nothing but division of the cell, right? So here in the different phases, the condensation of the chromosome that take place, and so these chromosomes they divide longitudinally to form 2 equal halves, to have 2 chromatids there, and the division of the chromosomes once again take place. So like that, all those activities and the formation of spindle fibers, alignment of the chromosomes that take place over here in this M phase itself, right? During the karyokinesis. taking place. During cytokinesis, what happens over here is equal division of the cell organelles as well as equal division of the cytoplasm that take place in this cytokinesis. How does it take place? How it differs in animal cell as well as plant cell, right? All those things that we study detailed manner in mitotic cell division as well as meiotic cell division. Apart from this one more phase I had explained, so that is G0 phase. What is this G0 phase? Very important one. G0 phase. So this is the resting period of the cell where the cell is not going to divide itself. So and it is not preparing itself for division itself, right? So that phase itself is called as G0 phase. So this occurs during early G1 phase. This occurs during early G1 phase. What happens over here is, so during early G1 phase, so the arrest of the cell division that occurs over here. Why? Because of the production of those proteins that stimulate the next phase, so are not produced there properly. Because of that one, so the cell growth that arrests there itself, and the cell will be at the rest and it does not prepare itself for division itself. That is G0 phase it is called as. So and in this phase, so cells are not metabolically active in nature, right? And they differentiate themselves into different types of mature cells. See here, so during G0 phase, some of the cells they differentiate into some cells like nerve cells. Some cells like nerve cells. If nerve cell is formed, it is not dividing itself. It does not divide itself. Alright? It is a matured cell. It starts to do one particular function. In the same way, the skeletal cell, skeletal muscle cell, it also does not divide. It starts to do one particular function. Means differentiate in the sense that, so the normal cell, so that convert itself into different shaped or permanent shaped cell, and this cell, what it formed is ultimately, what it formed? It formed ultimately the nerve cell. This is the normal cell which metamorphosis take place. Means what? So morphological change in structure that take place. So this cell may undergo division, but it arrests itself. So it differentiates into this cell and start to do one particular function. And this cell once again does not undergo division. Right? So that way these cells differentiate themselves and start to do one particular function. Right? One thing. Sometimes what happens, some of the cells so arrest themselves during early stage of the G1 phase, and they become metabolically inactive in nature. And once again they become metabolically active in nature, and they grow themselves and they divide themselves. They divide themselves, right? That kind of cells are also there. Say for example, in case of plants, permanent cells like parenchyma cells, they whenever they become permanent and they become matured and permanent, and they get one particular shape and start to do one particular function. But at the same time, if any damage on the body of these plants, that moment those parenchyma matured and permanent cells, once again they start to divide. Right? they regain the capacity to divide themselves. That possibility is also there itself, right? This is about the G0 phase. So now we came to know what is cell cycle, how does it occur. So everything we came to know. So now we are going to study different types of cell division in the next session. Thank you.